Hello and welcome to Nikolay's genetics lessons, and today I'm going to show you how to calculate colony forming units or CFU. And here's the problem. You conduct a standard plate count where 250 microliters of each dilution is added to each Petri plate. The Petri plates diluted to 10 to the power minus 2, 10 power minus 4 are Lowndes and cannot be counted. The 10 power minus 6 diluted Petri plate results in the 89 colonies. The dilution of 10 to the power minus 8 Petri plate has 3 colonies on it, so it cannot be used for CFU calculation since it is not within 25 to 150 colonies range. Calculate CFU and cells per milliliter of the original sample based on these results. Imagine that this is Petri dish and this is drop of water. I draw them out of scale, and in this drop of water we have some bacteria. Because it is so highly diluted that we don't have too many bacteria here, and when we put this drop of water on this Petri dish, each bacteria here would form a colony. So we have 5 bacteria here, so when we transfer them here, 1, 2, 3, 4, 5, each of them would form a colony. One bacteria we wouldn't be able to see with a naked eye, but a colony we would be able to see and we would be able to count them. So 1, 2, 3, 4, and 5. I also want to bring to your attention that colony forming units is not necessary means number of bacteria. For example, we may have actually in this drop of water more bacteria here, which also we will transfer here, but this bacteria is not viable and is not going to form a colony. So we wouldn't be able to see them with our naked eye. We also may have here some bacteria that are going to be dead. So this bacteria also is not going to form any colony here, and we wouldn't be able to see them, and we are not going to count them. So colony forming units means bacteria, but bacteria that forms colonies, so viable bacteria. Now take a look. We have to give an answer as cells per milliliter, but we start with 250 microliters. 1 milliliter equals to 1000 microliters. Actually, when we start with 250 microliters, in milliliters it's going to be 1/4 of a milliliter. Now also take a look. When we diluted our original sample 1 million times, we got 89 colonies, which is within this range, 25 to 250 colonies. Because we also got some colonies when we diluted to 10 power minus 8, which is 100 million times, but 3 colonies is not within this range, so we ignore this Petri dish. So we got 89 colonies, 89 colonies, but we started with 250 microliters, which is 1/4 of the milliliter. So we have to multiply by factor 4, and this is going to be 356 bacteria if we start not with 250 microliters, but with 1 milliliter. 10 power minus 6, 10 power minus 6 means dilution 1 million times. So 1 divided by 1 million. 1, 2, 3, 1, 2, 3. 1 divided by 1 million. That means that we have to multiply 356 by 1 million. So denominator here means dilution factor. So we have to multiply number of the colonies by dilution factor. And our answer is going to be 3, 5, 6, and 6 zeros. 1, 2, 3, 1, 2, 3. But usually we don't give such an answer. Usually 356 millions we convert into the scientific notation. Imagine that we have decimal point here because this is whole number. We have to move this decimal point to the left 1, 2, 3, 4, 5, 6, 7, 8 places. Why 8 places and not 7 or 6? Because in our scientific notation, number have to be between 1 and 10. So here we have 3.56 times 10 power 8. So what this number stands for? It stands for the colony forming units per 1 milliliter of the undiluted sample. And this is all for today. Subscribe and see you in the next video. Goodbye.